discussing about the sporophytic and the gametophytic self incompatibility, and I was explaining to you that it is the proteins which are present in the intine and in the exine which have a role to play in the incompatibility reaction. Now, in this one you can see that there is incorporation of proteins in the sporophytic system of incompatibility, and these proteins are contributed by the tapetum and they are present in the exine. Since they are in the exine, they are released immediately once the pollen lands on the surface of the stigma, and these proteins are readily available after hydration and they enable a speedy incompatibility reaction. This is as compared to the gametophytic self incompatibility systems where the proteins are present in the intine and they are released at a later stage because the first pollen needs to be hydrated, and of course the whole process takes little more time than the time taken in SSI systems. Sporophytic self incompatibility, here the pollen are generally shed at 3 cell stage and they have a very high metabolic rate and hence they are short lived. Recognition rejection reactions, they are very rapid, and this inhibition of pollen tube which occurs on the stigma, it can occur even within 30 seconds of the interaction or contact between the pollen and the stigma. The sporophytic self incompatible pollinations, they lead to the failure of pollen grain to germinate, or if the pollen grain germinates, there is formation of malformed pollen tubes which cease to penetrate the stigmatic tissue, or there can be emergence of multiple pollen tubes, or even the tip of the pollen tube can get swollen, or there can be formation of abortive pollen tubes. In case of any of these situations, there will be an incompatible reaction. Once the pollen grains are hydrated, once they land on the stigma and are hydrated, the proteins which are held in the exine are released and they interact with the proteins of the pellicle. So, this interaction leads to a recognition and rejection reaction. And the characteristic feature of this recognition rejection reaction is that in case the pollen has to be rejected, then there is development of callose plugs. Plug formation occurs at the point of contact of pollen grain with the stigma and also at the tip of the pollen tube. Now, the deposition of this callose is a consequence of the inhibition. Remember, it is not a cause of inhibition, it is because it has to be inhibited, and these callose plugs, they are going to inhibit the further growth of the pollen tubes, and it primarily occurs in those species which possess a dry type of stigma. You can see here that in figure A, pollen tube is able to penetrate the cuticle pellicle layer of stigmatic papilla, which is occurring in case of cross pollination. Now, this was a compatible pollination. In case the pollination is incompatible, then there is inhibition of this self pollen which results in the formation of a callose plug. That is figure B. It shows that the pollen is not able to germinate and there is formation of a callose plug. In Brassica, 3 S locus specific proteins are involved in incompatible pollen pistil interactions. One is SRK, that is S locus receptor kinase, other is SCR, S locus cysteine rich protein, also locus protein 11, and the third one is SLG, that is S locus glycoprotein. So, these 3 are primarily involved in incompatible pollen pistil interactions. In the rejection of self pollen, the S locus protein 11, it interacts with the stigmatic receptor kinase. This initiates a signaling cascade, that is, it sends in signals, that is, one after the other within the stigmatic papilla, and it finally conveys the message that the pollen has to be rejected. Gametophytic self-incompatibility is the one where the pollen are shed at the 2-cell stage. They exhibit a slow metabolic rate and hence they are long-lived. Germination of these grains is not constrained by the stigma, but pollen tube growth is inhibited in the style. Like SSI, callose leads to pollen tube growth arrest, and usually the plants which exhibit GSI, they possess a wet stigma, and peroxidase 10 plays a major role in the rejection reactions. In Lycopersicon peruvianum, pollen tubes have two layered cell walls. The outer is pectocellulosic and inner is callose. The pollen tubes, when they reach the incompatible styles after maybe traveling 2/3 of the styler length, they exhibit a very thin inner wall and numerous particles accumulating in the pollen tube cytoplasm. Eventually, this inner wall disappears and the pollen tube bursts. This bursting of the pollen tube is an indication of an incompatibility reaction. So, in Nicotiana, styles with incompatible tubes, they show higher activity of peroxidase 10, suggesting probable role in rejection. In Papaveracea, GSI is based on programmed cell death. So, whatever be the case, rejection can be due to disappearance of inner pollen wall. It can be because of higher activity of peroxidase 10 enzyme, or it could even be because of programmed cell death. Right. Now, I have talked to you about both SSI and GSI, and you have to remember that sporophytic self-incompatibility is the one which takes place immediately, that is, it can even occur within 30 seconds, whereas GSI is the one which takes longer, and hence the rejection reaction is occurring in the styler canal and not on the stigma, because the proteins involved in the gametophytic self-incompatibility, they are present in the intine, and after the hydration of pollen, they take a little longer to express themselves as compared to SSI, where there is an immediate reaction after hydration, right. And then we move to overcoming incompatibility. Now, we have so far been talking about the benefits of self-incompatibility, and I have told you that self-incompatibility promotes outbreeding, plays a pivotal role in evolution and diversification of angiosperms. This leads to plant improvement. Then why do we talk about overcoming this self-incompatibility? It is very important to overcome self-incompatibility because this will prevent the inbreeding of hybrids. In order to implement efficient breeding programs, we have to understand self-incompatibility systems, and it is very, very important at this stage to develop mechanisms which can break down the self-incompatibility. These are some of the methods which we use to overcome self-incompatibility: mixed pollination, bud pollination, stub pollination, intra-ovarian pollination. I would be taking each one of these methods briefly, and we start with mixed pollination. Now, mixed pollination means we are dusting the stigma with two types of pollen grains, one which are compatible and the second are those which are incompatible. Now, those pollen which are incompatible, they are not able to germinate on the surface of stigma. So, when we put the other type of pollen also which are compatible, then we term this as mixed pollination. This is adopted where pollen grains are unable to germinate due to absence of certain exine proteins. Stigma, when we dust with compatible as well as incompatible pollen together, the compatible pollen have the ability to germinate because they have the exine proteins which are required to for the pollen to germinate. So these are released once the pollen lands on the compatible stigma. Such pollen are now the compatible ones. They are inactivated by irradiation or by treating them with chemicals like methanol or other methods, so that they are not able to fertilize. These are known as the mentor pollen, and these are also called as the recognition pollen. Their role is only to promote the recognition reaction, and they allow the incompatible pollen to germinate. The recognition proteins from the wall of the mentor pollen help incompatible pollen to germinate on stigmas. Remember that these proteins are absent in incompatible pollen. They provide a pollen factor which is called as the P factor, P from pollen, which interacts with the S factor from the stigma, rendering it accessible to incompatible pollen. Mentor pollen also provide a growth promoting substance. It is called as the pollen growth promoting substance, which allows sustained growth of pollen tubes in the style. Now mind it, this pollen growth promoting substance is not only allowing the pollen tubes to grow, it is also providing substances critical for sustained growth of ovules, ovary and other fruit tissues, thus directly influencing fertilization events and also seed maturation. The second method we talk about is the intra-ovarian pollination. Now here you see that there is an ovary. This is an example of Papaver somniferum. Here what is done is that there are two punctures which are made in the ovary right opposite to each other. Right? This can only happen in those ovaries which are large enough, otherwise in small ovaries the process is very difficult. So what basically is done in intra-ovarian pollination is, intra means inside and ovarian is inside the ovary. So pollination is done inside the ovary. So we have actually excluded all the other stages of pollen landing on the stigma, germinating, moving through the styler canal. So directly the pollen are injected into the ovary. So pollination is accomplished by introducing a pollen suspension, and this pollen suspension is basically prepared by mixing pollen with distilled water and also with those substances which directly influence the germination of pollen. So they are directly poured into the ovary, and this method was devised by Kanta and Maheshwari in 1963 in Papaver somniferum. First of all, the surface of the ovary is sterilized with ethanol and two punctures are made opposite to each other. Pollen suspension is added with the help of a hypodermic syringe through one of the punctures. Now the second puncture is to allow the air to escape. Once pollination is accomplished, these punctures are sealed with petroleum jelly, and the technique, as you have seen, this is applicable only to species which have large ovaries. If we talk about Utricularia inflexa, where the ovary is very small, less than 1 millimeter, it becomes difficult to implement such kind of process. Then is in vitro pollination and fertilization. In vitro pollination and fertilization means that you are doing it in glass under laboratory conditions. So what is happening? The pollen grains are directly dusted on the ovules by completely removing all the stigmatic, styler and ovarian tissues. So you have the ovules which are put on a nutrient medium, and pollen are dusted on these ovules, and also this medium contains a pollen germination promoting substance. When pollination is done directly on the ovules under laboratory in vitro conditions, it is called ovular pollination, while pollination on ovules attached to placenta is known as the placental pollination. Now this type of in vitro pollination has proved beneficial in Petunia species where a normal seed set is observed. Now the major disadvantage of this method is that you cannot very easily and commonly do this, because there is requirement of an elaborate tissue culture room, tissue culture techniques and aseptic conditions. Only then such procedure can be followed. Now, this one is a picture which is taken from in vitro pollination in Papaver somniferum. The first one, or the A figure, you can see a portion of placenta with ovules. So, this is what placental pollination done. And then the second one is a pollinated culture showing pollen germination and pollen tube growth on and around the ovule. In the third one, you can see that the pollen tube is entering into the ovule. The last two, that is D and E, these are the different stages in the development of the pro-embryo. So, you can see a well defined pro-embryo in E. Now, I have talked to you about intra-ovarian pollination and also in vitro pollination and fertilization. The intra-ovarian pollination does not require any kind of tissue culture setup or aseptic conditions, whereas in vitro pollination and fertilization, we require these tissue culture techniques. And of course, for intra-ovarian pollination, the size of the ovary has to be large, and in vitro pollination and fertilization, it is not required, but there is requirement of a tissue culture medium in case of in vitro pollination and fertilization, which is not required in intra-ovarian pollination. Another method to overcome self-incompatibility is to modify the stigma surface. Now, there are many methods by means of which the surface of the stigma is modified. Now, this method is applied in species where the surface of stigma is a barrier for pollen to germinate. So, we have to modify the stigma. We can either use some solvents like hexane or detergents like Triton. There can be some chemicals like concanvalin A which can be applied on the surface of stigma, or even we can give heat treatment to the stigma in order to allow the pollen to germinate on the stigma surface. Concanvalin A is applied to stigma surface in Brassica. This concanvalin A will bind and mask the pellicle, and pellicle is the one which is responsible for incompatibility. If we use hexane or Triton X, they also have a similar mechanism of blocking the pellicle. And then there is application of calcium channel blockers on stigma, which allow pollen grains to grow the tubes down the style and to the ovary. And then there is heat treatment of style. Now, it has been reported that if you subject the styles of lilium to moderately high temperatures of 50 degrees and do it for up to 6 minutes prior to pollination, this will render self-pollen compatible. But if you raise the temperature above this, it will prove injurious to the pistil, and lowering temperature proves ineffective in overcoming incompatibility. So, possibly it is the heat treatment which inactivates the inhibitors of pollen. Then of course, there are other methods of irradiation and increased level of CO2. In irradiation, you are giving an X-ray treatment to the pollen, to the style, and this induces temporary breakdown of incompatibility reactions, right. And so, it will what will happen that X-ray can either cause unstable cytological changes in the pollen or style, or induce mutation in the incompatibility gene. And if we raise the level of CO2 in the vicinity of the stigma to about 100-fold at a relative humidity of 100% for several hours before pollination, this too will render incompatible pollen as pollen. And the last method for overcoming incompatibility is parasexual hybridization. This is also called as the somatic hybridization, where there is production of hybrids through fusion of isolated protoplasm of two different species. it overcomes the incompatibility which is encountered at either stigma, style or even ovary, was first developed by E.C. Cocking in 1969. Now, parasexual hybridization is basically the fusion of protoplasts. Protoplasts are also the naked cells. These are obtained through enzymatic degradation of the cell wall. So, once the cell wall is removed, the cell becomes naked and we call it as the protoplast, and two protoplasts are made to fuse. Bhojwani and Cocking in 1972 for the first time used protoplasts of microspores or pollen grains, and these were used because they are rich in cytoplasm and they readily fuse together. Being haploid, they are very important in hybridization. So, there are three major steps which are involved in the fusion of protoplasts. One is that you isolate the protoplast from whatever species you require. Okay. So, first is isolation of protoplasts, and once the protoplasts are isolated, in the second step these isolated protoplasts are fused together to create hybrid protoplasts. The last or the third step is culture of hybrid protoplasts to regenerate into whole plants. So, when we have to isolate protoplasts, what do? Cells are treated with a mixture of cellulase and pectinase enzymes. So, what do? They dissolve the cellulose and the pectin which is present in the cell wall matrix, and once they are dissolved, then of course we obtain naked protoplasts. Concentration of these enzymes, that is cellulase, pectinase, required for procedure is dependent on the tissue which is used, and it also varies from one species to the other species. Osmotic fragility is very important of isolated protoplasts, and this is the most crucial point in fusion of protoplasts, and we have to maintain this osmotic fragility right from enzyme solution to the washing solution, fusion and culture medium. Generally, a metabolically inert sugar like mannitol is used as an osmotic stabilizer. Freshly isolated protoplasts, in the presence of a suitable osmoticum, they appear spherical in shape. The second step is fusion of these isolated protoplasts. Now, fusion of these isolated protoplasts, this is done with the help of agents called as the fusogens. These are called fusogens because they help in the fusion of protoplasts. Polyethylene glycol, also called as PEG, is one such agent or a fusogen. It can fuse protoplasts from diverse taxa. You can fuse protoplasts from corn with soybean or pea, and it can even fuse a plant cell with an animal cell. Equal quantities of dense suspension of the two types of protoplasts are mixed along with 15% PEG solution. After about 10 to 30 minutes, this PEG solution or the PEG solution is diluted with the medium in which it was originally prepared, and it is finally washed off completely. Soon the protoplasts, they become closely pressed to each other over major portion of their membranes. Their cytoplasmic continuities, they expand and they fuse to become broader connections. So, you would remember that wherever there are plasmodesmatal connections, initially when they broaden up, they form channels through which all the cytoplasm can move around. So, eventually the protoplasts, they fuse completely and attain a spherical shape. Fusion of two dissimilar protoplasts leads to the formation of a heterokaryon. Okay. Now, this heterokaryon is the one which is formed, and the last step is the culture of these hybrid protoplasts. Now the hybrid protoplast is obtained by fusion of two protoplasts from two different species, and they are now put under culture conditions. Two nuclei of heterokaryon, they fuse to establish a true hybrid cell. The fused protoplasts, they synthesize a new wall around themselves. So, what we started with dissolving the walls, then we had naked protoplasts, we made the protoplasts fuse, and once they fused with each other, we have a hybrid protoplast. This hybrid protoplast ultimately it establishes a wall around itself, a new wall, and a new cell is reconstituted. These cells they divide and redivide to form a callus, which ultimately forms a plantlet. So, this is one of the most important methods of overcoming incompatibility, and I hope you have understood all the methods of overcoming self incompatibility. Thank you.